Welcome to The Deep Dive, the show where we take a stack of sources, articles, and research and distill them into the most important nuggets of knowledge, all custom-tailored for your curiosity. Today we're embarking on a journey that sounds like something straight out of science fiction, but it's a story that has profoundly shaped our past, our present, and will absolutely shape our future. I'm talking about plants changed by atomic energy. Imagine a world where the very food on your table, the crops that sustain billions, were transformed, not by traditional breeding, but by something as surprising and powerful as radiation.
It sounds almost futuristic, doesn't it? But this isn't just a concept, it's a tangible reality, a powerful force already at work. What we're diving into today is the fascinating field of induced plant mutations. It's a powerful, yet often overlooked tool in agricultural science. Think of it as an accelerated pathway to unlocking entirely new beneficial traits in crops, traits that are absolutely crucial for global food security, for improving nutrition, and, for adapting our food supply to the ever-increasing challenges of environmental change. It's truly a kind of secret ingredient in modern farming.
And that's precisely what we want to unravel. The big problem we're always trying to solve in agriculture, the core question really, is how to feed a growing population with better, more resilient food. So, our curiosity leads us to ask, what are the unknowns here? How did this seemingly radical idea, this connection between atomic power and plant breeding, become such a cornerstone of modern agriculture? What was the historical spark? How do scientists actually do it? How do they induce these subtle or sometimes dramatic changes in a plant's very DNA? What surprising successes emerged from these efforts, and what challenges still persist in this ongoing quest for agricultural innovation?
And to answer those unknowns, the data we have is pretty extensive. We've taken a deep dive into a whole collection of research papers, historical accounts, and cutting-edge scientific studies. This data allows us to explore the fundamental mechanisms of mutation, right down at the cellular level. We can examine specific, impactful examples across a wide range of crops and look at the sophisticated molecular techniques that continue to refine and advance this entire field.
Okay, so our mission today, what we want you to get out of this, is a shortcut to being truly well-informed about this incredible intersection of nuclear science, genetics, and agriculture. By the end of this deep dive, you're going to have a much deeper appreciation for the innovation, the perseverance, and, even the daring behind the very food that fills your plate. Get ready to have your curiosity challenged at every turn, and hopefully to desire even more solutions as we explore this surprising history. Let's get started. The atomic dawn of agriculture, a Cold War creation. So, to truly grasp the story of these atomic plants, we need to rewind. We need to go back to a time that might seem completely unrelated, the intense geopolitical climate of the 1950s. Imagine the world gripped by the palpable tension of the Cold War. When you think of that era, you probably picture air raid drills, missile silos, maybe intricate spy networks, right? But surprisingly, this was also a time when atomic science, born from the most destructive weapon ever created, began to blossom in an entirely different kind of field, our gardens and our agricultural lands. What's truly fascinating here, the real unknown for many, is how geopolitics directly fueled this agricultural research, pushing the boundaries of what was thought possible in food production.
That's absolutely key. The Cold War undeniably boosted the application of atomic science in agriculture and food. It started in the 1950s and happened across both sides of the Iron Curtain. Now, the earliest research into using radiation to affect plants actually goes way back, maybe late 19th century. And there were groundbreaking discoveries in nuclear physics and chemistry in the 1930s that paved the way. But it was truly the shadow of the atomic bomb, the most momentous wartime innovation combined with the intense political climate of the Cold War, that really set the stage for a magnified application of radioisotopes and ionizing radiation in agricultural research. This gave all materials, practices, and technologies that later became known as nuclear agriculture an intensely political significance. Superpowers on both sides understood that advancing nuclear literacy and gaining access to nuclear resources and technology was, well, a first-class power factor, a way to demonstrate scientific prowess and ensure national security through self-sufficiency in food.
And you can see this commitment reflected in very concrete actions, right? Even in a place like divided post-war Germany. For example, in West Germany, a significant turning point in their nuclear research and technology came in 1955. That year, the Ministry of Atomic Affairs was established. That's a pretty clear signal from Chancellor Konrad Adenauer that rapid inclusion in the atomic age was a national priority. And to push this forward, key figures like Johann Kuprianoff, who was the director of the Federal Research Institute for Food Preservation, and Wolfgang Flagg, director of the Institute for Soil Biochemistry, they were appointed as atomic advisors to the Federal Ministry for Nutrition, Agriculture and Forestry, the FMA. Flagg apparently had expressed interest in radioisotope research as early as the summer of 1955. He even went so far as to compile a comprehensive list of planned radioisotope research projects for his institute just a few months later in September of that year. This wasn't just theoretical talk, it was a strategically planned national endeavor, really showcasing political and scientific might.
Absolutely. And here's where it gets truly unexpected, maybe even a bit strange from our perspective. This wasn't all happening behind closed doors in secret labs. The whole concept of atomic agriculture captured the public imagination in a really powerful way. There was widespread publicity and incredibly enthusiastic claims surrounding this new technology. You can see this sentiment perfectly captured in an article from the New York Times in August 1955. The headline boldly proclaimed, "Irradiated seed will make the desert bloom." For fact, people genuinely believed that atomic energy held the key to solving the monumental problem of global hunger.
It sounds almost utopian, doesn't it? This idea that atomic power could just fix everything.
Okay.
But the most fascinating aspect, I think, was how this curiosity blossomed into this growing movement to bring atomic energy and experimentation directly into the lives of ordinary citizens. It wasn't just for government scientists anymore. Government subsidies were actually provided, allowing entrepreneurs and independent researchers to access nuclear sources and irradiate their own seeds. These irradiated seeds could then be sold to gardeners, school science clubs, even individual families to grow at home. And here's the kicker. Any interesting or unusual effects observed in these plants were encouraged to be reported back. This created what was essentially a nationwide effort of citizen science. A truly unique public participation in cutting-edge research, a real expression of curiosity blossoming beyond the lab walls.
And it wasn't just talk or enthusiastic projections. There's a very tangible, very popular success right from these early experiments, one that many of us probably encounter regularly without ever realizing its atomic origins. I'm talking about the ruby red grapefruit. This fruit is highlighted as one of the first and notably most commercially successful fruits to emerge from the so-called gamma gardens of this era. It was first cultivated in 1927 from seeds that were mutated with atomic radiation. Now, it's important to remember, human intervention in plant genetics has a long history, right? Grapefruits themselves, for instance, were first crossbred back in the 18th century from pomelos and sweet oranges. That was basically human-guided cross-pollination. But with atomic tools, scientists suddenly had this novel accelerated pathway to induce genetic changes and discover new desirable traits like that iconic deep red color. They were speeding up evolution in a very intentional way. Mechanics of mutation. Inducing genetic change.
Okay, that's a fascinating look at the historical spark, the why behind it all. But when we talk about atomic plants, what are we actually doing to them at a fundamental level? What's the data tell us about the biology? Are we literally scrambling their DNA? And how does a plant even survive that kind of radical intervention?
You've hit on the core of it. While mutations are fundamental to life, absolutely essential for evolution and genetic diversity, they are fundamentally caused by damage to DNA, the blueprint of life. Our sources explain that understanding the different ways DNA can be damaged, and crucially, how the cell attempts to repair it, is critical. It's not just about the damage, but the repair too. For instance, gamma irradiation, a common method we mentioned, often leads to what we call DNA double-strand breaks, or DSBs. Think of it like a ladder being cut completely in half. Ultraviolet or UV radiation, however, works differently. It often causes two DNA building blocks right next to each other to abnormally stick together, like a tiny kink or bubble in the DNA ladder. And then you have chemical mutagens. These usually cause misspellings in the DNA code, like typos, or they trigger the removal of entire sections. So each mutagen creates a distinct type of molecular injury.
So the plant's own repair mechanisms become a crucial part of the story here. It's not just about breaking the DNA, but how the plant tries to put it back together, and whether it does it perfectly or maybe with a beneficial typo.
Exactly, that's the crux. When a double-strand break happens, for example, there are two main repair pathways. One is called homologous recombination, or HR. This is like a skilled carpenter who has the exact blueprint and perfectly repairs the broken ladder. It uses a template, usually the identical copy of the DNA strand, to perfectly fix the break. This results in very few, if any, mutations, very precise. However, the other pathway is non-homologous end joining, or NHE. This process is inherently error-prone. Think of it more like someone rushing to fix the broken ladder with super glue. They just jam the broken ends back together. This often leads to small insertions or deletions, little mistakes, and thus, it effectively produces the very mutations we're often looking for. Understanding these intricate mechanisms is very important for properly designing mutagenic experiments. You want to achieve an enhanced mutation frequency. For example, if you use a genetic line that is defective in that precise HR repair pathway, or if you use haploid materials like pollen or anther cells, which lack that matching DNA template for HR, while you would expect to produce a significantly higher frequency of mutations after radiation treatment because the plant is essentially forced to rely more on the error-prone NHEJ pathway. This increases the chances of getting a useful typo.
Okay, so we've just established the biological why of mutations, the DNA damage and repair part. Now let's explore the how. The early days of induced mutation relied on a few key methods, some of which, as you said, sound almost like something out of a classic sci-fi film.
They certainly do. One of the most iconic methods was the use of gamma gardens, or gamma fields. Picture this. It's typically a large circular area of land, sometimes several acres in size. At the very center, there's a powerful shielded radiation source, often cobalt-60. Plants are arranged in concentric circles around this source. Now imagine walking into this living laboratory. The plants closest to the central radiation source, they would generally die. Too much radiation. Further out, many plants would be severely stunted or they develop tumors, showing obvious signs of extreme radiation damage. But critically, it was around the edges of this affected zone, where the radiation dose was much lower and chronic, that many plants looked relatively normal. It was here that researchers would meticulously examine tens of thousands of individual plants, looking for the ones with beneficial mutations, maybe brighter fruit color, bigger flowers, or better resistance to cold. This chronic irradiation approach was designed to expose plants to continuous low-dose gamma rays over time. This allowed mutated cells to emerge and accumulate mutations in growing organs, unlike a single acute high dose. It was a painstaking search for those lucky accidents, those beneficial mutations.
So it was like a living experiment, mapping the effects of radiation intensity onto plant development in real time. Beyond these impressive, almost eerie radiation fields, what other traditional methods were employed to induce mutations in those early days?
Alongside physical mutagens like gamma rays, chemical mutagens played a significant role. Ethyl methensulfonate, or EMS, for example, has been and still is commonly used. Why? Because it leads to irreversible changes in the DNA with a very high frequency. This characteristic makes it capable of generating what we call saturated mutagenized populations with a relatively small number of individuals. It makes the screening process more efficient for finding certain types of genetic changes. Another chemical mutagen, sodium azide, or SA, was notably used for seed treatment in barley, particularly for what's called forward genetic screening, where you induce mutations and then look for specific desired traits. Now, a crucial aspect of both radiation and chemical mutagenesis is dose consideration. It's really important. It's not about maximum exposure. It's about finding that sweet spot, the right dose that causes enough mutations without killing too many plants. For instance, rice typically tolerates doses between 200 to 300 grays. That's the unit of absorbed radiation dose. A crop like kiwicha can handle maybe around 400 grays. For pineapple, the lethal dose for 50% of the population, the LD50, for shoot formation is around 40 grays. And for 100% lethality, LD100, it's about 83 grays. The ideal dose allows for a high frequency of mutations without causing excessive lethality, ensuring you have enough viable plants left to select from.
Okay, so chemical baths and vast radiation fields already sound intense. But imagine sending seeds into space. This takes the concept of extreme environments for mutation to a whole new level, literally harnessing cosmic forces to unlock genetic potential.
It's an incredibly bold approach, yeah. And one that China, in particular, has invested heavily in. The aerospace environment is truly unique. You've got strong cosmic radiation, persistent microgravity, weak geomagnetic fields, and this hyper-clean super vacuum. A whole cocktail of stresses. Extensive experimental data has clearly shown that these space conditions dramatically affect plant growth and development. And crucially, they induce significant genetic changes in crop seeds. For example, the rate of cell division, mitosis, declines remarkably in space-flown seeds, and the type and frequency of chromosomal errors, like breaks or rearrangements, are greatly increased compared to the controls left on Earth. What's fascinating is that these mutagenic effects appear to be universal among plants. It's not just one type. The combined effects of cosmic radiation and microgravity, along with other spaceflight factors, seem to be the primary drivers of these genetic changes. It's almost like a natural, high-powered mutagen up there.
So it's not just one stressor, but a combination of these extreme conditions causing the mutations. Can you tell us more about China's pioneering work here? They seem to be leading the way.
They really do have a robust space breeding program. Since 1987, they have systematically conducted experiments using various platforms. They've used recoverable satellites, their Shenzhou spacecraft, and even high-altitude balloons. A notable milestone was the Shijian 8 satellite, launched back in 2006. This was actually the first satellite in the world specifically designed for a space breeding program, and it carried over 2,000 plant accessions. That's samples of plant material from 133 different species into orbit. That's a huge payload of potential mutations. The results have been quite tangible. To date, 66 new mutant varieties of various crops, including staple foods like rice and wheat, but also cotton, rapeseed, sesame, pepper, tomato, and alfalfa, have been developed through this space breeding program and officially released in China. They have also obtained a valuable catalog of useful, rare mutant germplasm. This provides a unique resource for future breeding efforts, stuff they wouldn't easily find on Earth.
And are there any specific examples where we can really see the visual impact of these space-induced mutations, something concrete?
Absolutely. One excellent illustration comes from Spirulina platensis mutants. That's a type of blue-green algae. If you were to look at them under a microscope, the differences compared to their original Earthbound counterparts are quite striking. Some of the space-mutated Spirulina looked regular and robust, maybe even healthier, but others were linear and weak. Fascinatingly, some mutants were 2 to 3 times longer than the original Spirulina. This visual evidence powerfully demonstrates how direct and impactful space-induced mutation can be on a cellular and morphological level. It's creating entirely new forms right before your eyes.
Okay, so we've gone from these broad-stroke methods like radiation fields and space travel to now increasingly precise genetic sculpting. The toolkit for inducing mutations has become incredibly sophisticated. We're moving towards almost surgical precision in genetic modification.
Indeed, the technology has advanced remarkably. One such advanced technique involves using ion beams. Ion beams exhibit a high relative biological effectiveness, RBE, in causing things like lethality and inducing mutations, compared to conventional low LET radiation like gamma rays or X-rays. The mechanism behind this is that ion beams deposit a very high amount of energy onto a very localized target within the cell or DNA. This action primarily induces single or double-strand DNA breaks, but critically, these breaks often come with damaged ends that are much more difficult for the cell's natural repair mechanisms to fix easily. They're mesio breaks. As a result, ion beams frequently produce large-scale DNA alterations, things like inversions, where a segment flips around, translocations, where parts swap between chromosomes, and large deletions. They're better at causing big rearrangements rather than just small point mutations.
And is this just theoretical science, or are these ion beams actually being used right now to create new varieties? Are they making a real impact?
Oh, absolutely. Japan, for example, has been at the forefront of this research. Since 1991, their Takasaki Ion Accelerator Advanced Radiation Application Facility, known as TIARA, has consistently and efficiently produced novel mutants and varieties using ion beams. For instance, studies showed that a heavy ion beam treatment could induce significant mutations in wheat, primarily affecting things like the shape of the spike, the part with the grains, and the overall plant height. They even identified an optimal irradiation dose for maximizing these specific changes. This level of precision allows for the generation of very specific, large-scale genomic rearrangements that might be difficult or just impossible to achieve through other methods.
That's a huge leap from just exposing a whole field to gamma rays. It's much more targeted. What other precision tools are redefining the landscape of mutation breeding?
Another groundbreaking method is tilling. That stands for targeting induced local lesions in genomes. You can think of tilling like a super precise spell checker for DNA. It's a robust, high throughput method. And importantly, it's non-transgenic, meaning it doesn't involve inserting foreign DNA. It can be applied to most species. Its primary use is to identify single nucleotide mutations, single letter typos in the DNA code, and small insertions or deletions known as indels. The process itself is quite ingenious. Basically, you target a specific sentence, a gene of interest, and then you use special molecular tools, almost like tiny enzymatic scissors, that snip the DNA only where there's a mismatch, a single letter typo, between a normal copy and a mutated copy. By seeing where these snips happen and measuring the fragment sizes, you can pinpoint the location of the mutation within about 10 base pairs. Once you've found a potential mutation this way, you can then confirm the exact nucleotide change by sequencing that specific region of DNA. This allows researchers to find subtle, potentially beneficial changes much more efficiently than just screening thousands of plants visually.
That sounds incredibly precise, almost like finding a needle in a haystack, but with a really good map. Is there anything even more targeted than tilling? Can we edit a specific gene directly?
Yes, moving even further into direct genetic sculpting, we have technologies like zinc finger nucleases, or ZFNs. ZFNs are a method that uses engineered proteins, the zinc fingers, to guide molecular scissors nucleases to a very specific location in the genome. The key advantage here is that a specific gene can be targeted for mutation in situ. That means directly within its natural chromosomal location, leaving the rest of the genome largely untouched. This strategy avoids the risks often associated with older transgenic approaches, like unintended mutations arising from the random insertion of new DNA somewhere else in the genome. Instead, CFNs aim to make changes, or even incorporate new DNA sequences, at a predetermined precise site in the chromosome. This also ensures that the targeted gene, even if modified, remains under its normal endogenous controls within the plant. And as a proof of concept, studies using specific plant cell cultures have clearly confirmed successful ZFN-mediated gene targeting, showing that precise genetic changes could be made exactly as predicted. This level of control opens up incredible possibilities for precise genetic engineering, allowing us to edit the genome almost like editing a document.
Okay, so when we look back, let's check our result here. We've journeyed from the broad strokes of gamma radiation affecting entire fields, kind of a scattergun approach, to the incredible precision of molecular tools like tilling and zinc finger nucleases, which can target individual genes or even specific DNA bases. We've also seen how space itself can be a mutagen, which is just wild. This progression reflects a dramatic shift, really, from relying on random chance to embracing direct design.
Exactly. Early methods were like casting a wide net, hoping for a beneficial accident, a lucky mutation. Now we're increasingly using surgical precision, fundamentally accelerating and hopefully de-risking the breeding process. It's a profound evolution in how we understand and manipulate plant genetics.
Right. Each method offers different advantages and challenges. It depends on whether the goal is to create broad genetic diversity to screen from or to engineer a very specific known change into a crop. Funny trait, global collaboration and impact, enhancing food security and crop quality. So this whole field isn't just about individual labs making fascinating discoveries in isolation, is it? It sounds like a massive, coordinated global effort. And leading the charge, apparently, is a collaboration you might not immediately expect, bringing together two major international organizations.
You're likely referring to the joint FAO-IAEA program on nuclear techniques in food and agriculture. FAO being the Food and Agriculture Organization of the UN, and IAEA, the International Atomic Energy Agency. This initiative stands as a hugely successful model of cooperation within the United Nations system. Its fundamental purpose is to provide essential assistance to member states in the peaceful application of nuclear techniques, including induced mutation for improving food and agriculture systems worldwide. This program focuses its high-priority activities across three critical thematic areas. First, productivity enhancement, basically getting more food from the same land. Second, plant, animal, and consumer protection, ensuring the health and safety of our food supply. And third, the conservation and sustainable use of natural resources, which is absolutely vital for long-term agricultural viability.
That sounds incredibly comprehensive, a really broad mandate. How does it actually deliver on these goals and reach so many places around the world?
Well, it employs a very integrated approach. It encompasses policy advice, rigorous research and development, R&D, building capacity through training and knowledge transfer, and providing both normative setting standards and operational technical support directly to member states. The scale and reach of this program are truly impressive. Through IAEA technical cooperation funding, the joint FAO-IEA program provides technical support to more than 250 projects every single year. This includes sending experts, providing training, and helping procure essential equipment for these technical cooperation projects on the ground. Additionally, something like 400 to 500 institutions and experimental stations across member countries cooperate in 30 to 40 coordinated research projects annually, all organized by this joint program. This immense, structured global effort really underscores the critical importance placed on induced mutations as a tool for agricultural improvement on an international scale. It shows how international collaboration is tackling fundamental global challenges in food security.
Okay, so there's this huge global network. Now, beyond just growing more food, a critical aspect of food security is growing better food that is more nutritious and healthier. And mutations have been instrumental in making our staple crops more nutritionally valuable, addressing some really significant deficiencies. So the key insight here, deriving differently perhaps, isn't just about boosting a single nutrient. It's about engineering resilience against hidden hunger, fundamentally transforming staple crops from mere calories into powerful nutritional delivery systems, especially in vulnerable regions.
That's exactly right. One major area of success is the development of low phytic acid, or LPA, mutants. Now, the problem is that about 75%, sometimes even more, of the total phosphorus in seeds is found locked up as phytic acid. While phosphorus is an essential nutrient, phytic acid largely makes it unavailable for absorption by humans and non-ruminant animals like pigs and poultry. So imagine a grain of wheat or maize. In a normal grain, about 80% of the phosphorus is locked away, unusable by us. But with these low phytic acid mutants, that can dramatically drop to just around 27% unavailable. It's like unlocking a hidden pantry of nutrients within the grain itself. This lack of availability can lead to mineral deficiencies, particularly zinc and iron, in diets heavily reliant on cereals, and it also contributes to environmental pollution from excess phosphorus excreted in animal waste, which causes problems like algal blooms.
So the mutation essentially unlocks that phosphorus and potentially other minerals bound by the phytic acid.
Precisely. Mutations that specifically block the synthesis or accumulation of phytic acid during seed development have been successfully isolated in major crops like maize, barley, rice, wheat, and soybean. The benefits are really twofold, maybe even threefold. These mutations significantly reduce phosphorus loads going into agricultural lands and surface waters, which is a huge environmental plus. They also provide enhanced nutrition and metabolism for poultry and swine because the animals can absorb more phosphorus directly from their feed, reducing the need for expensive supplements. And for humans, it increases the bioavailability of essential minerals like iron and zinc. In barley, for example, specific recessive mutations can block seed phytic acid accumulation by 50% and reduce total seed phosphorus by 15%, all with little negative impact on yield. Researchers use advanced techniques like metabolite profiling to compare mutants and wild types. This allows them to pinpoint the exact genetic targets for these LPA mutations within the complex biochemical pathways that build phytic acid, giving us deeper insights into how to make these nutritional improvements even more effectively.
That's incredible. A mutation with environmental and nutritional benefits. What about improving the protein quality of staple crops? That's another huge issue.
That brings us to quality protein maize, or QPM. The challenge with normal maize endosperm protein is that it's notoriously deficient in two essential amino acids, lysine and tryptophan. These are amino acids our bodies can't produce themselves, so we absolutely must get them from our diet. The solution came with the discovery of the opaque-2 gene mutation and its strategic use in breeding programs, combined with other genetic modifier genes that improve the kernel texture and overall quality. This led to the development of QPM. The impact has been dramatic. QPM contains almost double the amount of lysine and tryptophan compared to normal maize. This leads to significant improvements in human and animal nutrition, growth, and overall performance, particularly in regions of the world where maize is a major dietary staple and protein deficiency is common.
So it's a huge nutritional upgrade, potentially life-changing in some areas. But is breeding QPM straightforward? Is it easy to get this trait into farmers' fields?
Not conventionally, no. The original opaque-2 gene is recessive, meaning a plant needs two copies to show the trait. And the modifier genes that contribute to the improved kernel quality are polygenic, meaning multiple genes are involved, which complicates things. This makes conventional breeding approaches quite tedious and time-consuming. It requires many generations of crossing and self-pollination, plus rigorous biochemical tests at each stage to ensure the enhanced lysine and tryptophan levels and the right kernel texture are consistently present. However, molecular solutions are accelerating this process significantly. Molecular markers, think of them as genetic signposts located within or very close to the opaque-2 gene, now provide opportunities for what's called marker-assisted selection, or AMS. This allows breeders to identify plants carrying the desired genes much more rapidly and efficiently, often at the seedling stage, without needing extensive phenotypic or biochemical assays later on. Institutions like Simorti, the International Maize and Wheat Improvement Center, are actively developing reliable, easy-to-use markers for key traits like endosperm hardness and free amino acid content. This is greatly speeding up QPM development and deployment programs, effectively giving breeders a valuable shortcut.
That's a powerful approach, using genetics to streamline breeding. What about other cereals, like barley? Has similar progress been made there?
High lysine barley is another significant story, yes. Back in the 1960s, there was major concern globally regarding the low nutritional quality of cereal protein as a food source, particularly for children, with lysine being the primary limiting amino acid in most cereals. In 1964, a new screening method was developed based on dye binding capacity, DBC, which could assay for basic amino acids like lysine relatively quickly. This breakthrough led, in 1966, to the identification of the first high lysine gene in an Ethiopian barley cultivar called Hipperly. Building on this, Hans Dahl's group in Denmark at the Rising National Nuclear Laboratory, known for its atomic research, extensively used this DBC method in the 1970s, screening mutagenized barley populations. They successfully isolated about 25 barley mutants with moderate to high lysine content in their protein. Now, these mutants often had lower starch content and sometimes lower yields, but they were crucial discoveries nonetheless. The most radical mutant they found was Rhizia 1508. It had a remarkable 45% increase in lysine and a biological value, a measure of protein quality almost equivalent to milk proteins. The impact was dramatically illustrated in a pig trial. Pigs fed Rhizia 1508 showed an 84% increase in growth over 3 months compared to pigs fed normal barley. It reduced the amount of barley consumed to reach slaughter weight from 538 kilograms for normal barley down to just 209 kilograms for the Rhizia 1508 unit, all without needing extra protein additives in the feed.
That's an astonishing improvement in feed efficiency, just from a single mutation. How are these mutants analyzed today? Do they still rely on those older methods?
Modern analysis utilizes much more sophisticated techniques like near-infrared reflectance or NIR spectral analysis combined with advanced statistical methods called chemometrics. Imagine shining a specific type of light through a ground barley sample and analyzing the unique spectral fingerprint that reflects back or passes through. These methods can clearly differentiate normal barley from high lysine mutants and recombinant lines based on their unique spectral profiles. You can literally see the difference in the light patterns. This allows for rapid, non-destructive screening and characterization of these valuable traits, significantly speeding up the breeding process compared to the older, slower biochemical assays.
Okay, so beyond general nutrition enhancement, induced mutations are even tackling very specific dietary challenges, like creating wheat that could potentially be safe for celiac patients. This is huge for people who suffer from this autoimmune disease, potentially offering a truly life-changing alternative to strict avoidance.
It truly is a significant area of research. For celiac patients, wheat, barley, and rye are typically off limits because of an autoimmune reaction against specific protein fragments, peptides, found mainly in proteins called gliadins. These peptides, which are rich in proline and glutamine amino acids, trigger an immune response in susceptible individuals that leads to chronic damage to the intestinal lining. The conventional solution is complete, lifelong dietary exclusion. However, a fascinating approach using mutation breeding focuses specifically on modifying or reducing these gliadin-type proteins. It's important to note that gliadins, while contributing to some DORT properties, are not absolutely essential for baking quality. Wheat's high molecular weight gluten proteins alone have been shown to be sufficient to produce high-quality breads.
So if they aren't essential for baking, can they be selectively reduced or even eliminated through mutation?
Precisely. That's the strategy. The genes that make gliadin-type proteins are normally silenced or turned off in the leaves and stems of the plant, but they get unsilenced or activated during grain development to produce the protein that ends up in the flour. The insight here is that if we can prevent that unsilencing process, specifically through mutation, we can inhibit the synthesis of these problematic gliadin-type proteins in the grain. As a proof of principle, a barley cultivar named Leciba already exists that has such a mutation, resulting in lower levels of similar proteins. This provides a clear rationale for creating wheat varieties by mutating the specific enzymes responsible for this unsilencing process in the endosperm. The goal is to ultimately offer celiac-safe wheat options that are much more palatable and easier to incorporate into a diet than many current gluten-free alternatives.
Right, moving beyond the basics of just increasing yield and boosting general nutritional value like protein. Mutations are also refining the very composition of our food, right down to the oils we consume, making them healthier, maybe more stable, more versatile for various applications.
That's right. In the realm of oil quality, significant advancements have been made through induced mutation. Take sunflower oil, for example. While already appreciated as a high-quality commodity, new emerging markets and increasing health concerns are demanding specific oil qualities. The optimal quality really depends heavily on the intended use. For instance, applications demanding high nutritional value, like salad oil, require a reduction in saturated fatty acids and maybe an enhancement of vitamin E, alpha-tocopherol. Conversely, uses in the food industry requiring plastic fats, like margarines or shortenings, actually benefit from an increased concentration of certain saturated fatty acids to improve texture and avoid the need for artificial hydrogenation, which produces trans fats. And for high-temperature processes like frying, or even for industrial uses like biolubricants, oils need to be highly resistant to breaking down under heat. They need thermo-oxidation resistance. This often requires a high concentration of monounsaturated oleic acid and natural antioxidants like gamma and delta tocopherols.
So how has mutagenesis helped tailor sunflower oil to these diverse needs? It sounds like you need different mutations for different end goals.
Exactly. In sunflower, mutagenesis has proven to be the most successful procedure to generate this kind of specific genetic variability for these oil quality components. The mutagenic treatment is usually applied to the seeds. Then, mutants are quickly detected by analyzing half seeds from the second generation M2 plants using methods like gas chromatography. This allows for rapid identification of promising lines within a single year. The most valuable sunflower oil quality mutants produced through this method include those with very high oleic acid content, often exceeding 80%, or high levels of specific saturated fatty acids like palmitic or stearic acid, sometimes above 25%. Mutants with very low total saturated fatty acids below 7% have also been developed for health markets, and others have increased levels of specific vitamin E forms to tocopherols, sometimes by over 95%, enhancing shelf life and nutritional value. Crucially, these novel traits are, in almost all cases, governed by a small number of genes, often just one or two. This significantly facilitates their management and incorporation into elite plant breeding programs. This induced variation truly opens up the possibility of tailoring specialty sunflower oils for very specific food and non-food applications. And just to help visualize the structure we are discussing here, imagine a simple glycerol molecule, which is like a 3-carbon backbone. Attached to each of these carbons is a long chain called a fatty acid. The type and arrangement of these 3 fatty acid chains dictate the oil's properties, whether it's liquid or solid, stable, or prone to oxidation. It's these fatty acids that are being precisely modified through mutation breeding.
That's a great illustration of the molecular changes driving the functional differences. What about other important oilseed crops, like groundnuts, peanuts, as we usually call them?
Groundnut production and quality have seen immense benefits, particularly through research conducted at the Bhabha Atomic Research Center, or BRRC, in Mumbai, India. They've been doing mutation research extensively there for the past half-century. Their impact has been vast. Over 100 new varieties of oilseeds and legumes, including groundnut, have been developed in India by incorporating desirable traits induced by mutation. These traits include things like large seed size, a semi-dwarf growth habit, which helps prevent lodging, high harvest index, meaning more pods relative to leaves and stems, better nutrient partitioning, fresh seed dormancy, important for storage, specific seed colors, drought tolerance, and resistance to various devastating diseases like powdery mildew, yellow mosaic virus, and bacterial pustule diseases. Many national and state breeding programs in India have successfully utilized these BRRC mutant varieties as parental materials, leading to the development and release of numerous improved varieties across the country.
So the impact is truly reaching farmers on the ground. It's making a tangible difference to their livelihoods and national production.
Absolutely. Many of these varieties have gained high patronage from farming communities and are extensively grown across India. Groundnut varieties developed through mutation breeding, in particular, have made a considerable impact by consistently yielding record harvests in farmers' fields. In major groundnut-producing states, productivity has almost doubled compared to older varieties. Hundreds of farmers are now harvesting significantly improved yields, sometimes up to 7 tons per hectare, earning a much better net profit, maybe up to $1,200 per hectare, which is substantial. Some progressive farmers, using optimal cultivation practices, have even achieved record yields of over 10 tons per hectare of dry pods, with specific mutant varieties like TAG 24 and TG 26. It's even reported, and this is quite remarkable, that groundnut was successfully cultivated at an altitude of 3,505 meters in Leh, Ladakh, using polythene mulch technology. This might be the world's first successful report of groundnut cultivation at that extreme altitude, demonstrating the incredible versatility and resilience that these induced mutations can confer.
That's amazing adaptability. And has this success in improving oil quality and yield been replicated in other oil crops besides sunflower and groundnut?
Yes, definitely. Mutation breeding has also significantly improved oil quality in soybean. For instance, high oleic acid mutants in soybean were induced by X-ray irradiation, offering a healthier oil profile. Similarly, in India, there's been successful development of Brassica napus canola quality varieties. That's a type of rapeseed specifically suited for Indian agroclimatic conditions, again achieved through induced mutations. Notable mutants showed significantly higher yields compared to conventional Czech varieties, and one particular mutant line possessed an impressive 46% oil content on average compared to 42% in its parent variety. This really shows how precise and impactful these induced mutations can be in optimizing oil composition and content for various market demands and health considerations across a range of important oilseed crops. Okay, let's take a moment here, maybe do a mega recap of what we've covered so far in sections 1 and 2. It's been quite a journey. We started with the fascinating historical context, seeing how the geopolitical tensions of the Cold War surprisingly spurred this agricultural revolution. This led to unexpected public enthusiasm, remember. Irradiated seed will make the desert bloom, and early tangible successes like the ruby red grapefruit. Then we dove deep into the science, the mechanics of mutation induction. We explored the methods from the broad, almost sci-fi concepts of gamma fields and chemical mutagens to the truly out-of-this-world idea of space-induced mutagenesis using cosmic rays. And we finished that section with the ultra-precise molecular tools like ion beams, tilling, and zinc finger nucleases. And most recently, we've just detailed the immense global impact driven by collaborations like the FAO-IAEA program. We focused on how mutations enhance food security by boosting the nutritional value of staple crops, improving protein quality with QPM and high lysine barley, tackling phytic acid issues, even exploring celiac-safe wheat. And we also covered the refinement of oil quality in crops like sunflower and groundnut, making them healthier and more versatile.
It's a powerful narrative of ingenuity, driven by curiosity and the need for solutions. As you, our listener, check the result of this journey so far. Think about how these historical drivers, the scientific breakthroughs, and the global collaborations all synergize. It's not just one factor, but a complex interplay that has propelled this field forward, fundamentally changing how we grow and think about our food. Maybe take a moment for a mental flashcard. Key terms we fit: Cold War agriculture, gamma gardens, ruby red grapefruit, DNA double-strand breaks, homologous recombination, HR, non-homologous end joining, NHEJ, EMS, ethyl methensulfonate, ion beams, space-induced mutagenesis, tilling, zinc finger nucleases, DFNs, the FAO-IAEA program, low phytic acid LPA, quality protein maize, QPM, high lysine barley, celiac-safe wheat potential, oil quality improvement, and that cool early citizen science aspect. Got those. Okay, let's move on. Diversifying applications from disease resistance to ornamental traits. So as our planet faces increasingly rapid changes, our crops are confronting new and intensifying challenges for the environment itself. Droughts are more frequent and severe in many places. Salinity is encroaching on fertile coastal and irrigated lands, and extreme temperatures, both heat and cold, are becoming the norm rather than the exception. In this context, induced mutations offer a really powerful avenue to build resilience directly into our food supply, making the plants themselves inherently tougher against these non-living or abiotic stresses.
Exactly. Induced mutations are equipping crops to better withstand these abiotic stresses, environmental factors that aren't living organisms, like pests or diseases, but which severely limit plant growth and yield. This is absolutely crucial for adapting agriculture to the realities of climate change. Let's take cowpea, for example. Cowpea is a vital staple crop, especially in many parts of Africa and Asia. One project we looked at aimed specifically to select mutant cowpea plants with improved levels of drought tolerance. A key goal was to do this without altering their seed coat color or overall growth form, because those are important traits for market acceptance by farmers and consumers. Early screening for these drought-tolerant traits was done simply at the seedling stage in wooden boxes. Then, mature plants were screened under more controlled conditions in rainout shelters, basically structures that prevent rain from reaching the plants, allowing researchers to precisely control water stress and simulate drought conditions accurately. Through this process, researchers identified specific physiological traits, like changes in leaf water potential or photosynthetic rate, that correlated with drought stress tolerance. They also found that variations in root structure, deeper or more extensive root systems, were linked to better performance under drought.
So it's about seeing how the plant changes its basic physiology, its structure, to cope with the lack of water.
What about the molecular insights? Did they look at the genes involved?
Yes, they did. At the molecular level, scientists used techniques like cDNA amplified fragment length polymorphism, cDNA-AFLP, to identify which genes were being turned on or off in response to drought stress at different time points. They found that different genes were expressed as the drought progressed. When they sequenced some of these fragments, they found high similarity to known genes in soybean, another legume. Many of these genes had possible functions in cell defense or resistance mechanisms, and importantly, in signal transduction, how the plant communicates internally about the stress it's experiencing. Further genetic studies using RAPD markers also showed clear genetic variation between the drought-tolerant mutant lines and the original control lines, confirming that genetic changes were indeed responsible for the improved tolerance. The outcome of this research was the identification of several promising mutant lines. The long-term plan is then to introgress or breed this drought tolerance trait from the best mutant line into existing, popular, but drought-susceptible South African cowpea cultivars, particularly those grown by resource-poor farmers who are most vulnerable to climate variability.
That's a great example for drought. Now, beyond drought, salinity is another massive agricultural challenge, especially with increasing irrigation, which can bring salts up, and saltwater intrusion into coastal areas due to sea level rise. Can mutations help plants deal with salty soil?
Yes, sugarcane provides a compelling example for developing salt tolerance. Salinity is a significant environmental stressor that drastically reduces plant productivity worldwide. To address this in sugarcane, scientists used in vitro mutagenesis. This involves taking small pieces of sugarcane tissue, growing them in the lab, in vitro, exposing these young embryogenic cultures to gamma radiation, typically doses from 10 to 50 gray, and then growing the resulting plantlets in media containing increasingly high levels of salt, specifically sodium chloride and NaCl. They observed that under salt stress, the normal, non-mutated sugarcane tissues grew more slowly and experienced more cell damage. However, the salt-tolerant mutant lines that survived the selection process were able to maintain better water status inside their cells, and crucially, they maintained a high potassium to sodium ratio. This indicated that these tolerant sugarcane lines can act as sodium excluders. They have mechanisms to actively prevent excessive sodium from entering their root cells, where they efficiently transport it out, almost like having a specialized filter system at the root level. From this whole process, a total of 147 potentially salt-tolerant plantlets were successfully selected at different salt levels. Molecular analysis using RAPD markers confirmed genetic differences, polymorphism among these selected lines compared to the original variety. Importantly, some of these salt-tolerant variants also showed desirable agronomic traits like improved cane yield and sucrose content, making them potentially very valuable for cultivation in saline-affected areas.
That's a clear pathway to more resilient sugarcane. How does salt tolerance work in staple grains like wheat? Is it a similar mechanism?
In wheat, the mechanisms can be complex, but one of the major strategies conferring salt tolerance is indeed sodium exclusion, particularly keeping sodium out of the sensitive leaves where photosynthesis occurs. This is often achieved by very low net uptake of sodium by the cells in the root cortex, the outer layers, and then tightly controlled transport of sodium away from the xylem, the water transport system, by specialized cells in the root's core. Some bread wheat cultivars naturally possess an enhanced potassium to sodium discrimination trait, often linked to a gene locus called Na1 on chromosome 4DL, which helps them cope better with salty conditions. Interestingly, durum wheat, which is used for pasta and is generally more salt-sensitive than bread wheat, lacks a direct equivalent of this Na1 gene. Genetic analysis in durum wheat showed that its sodium exclusion ability was controlled by two different dominant genes, NAX1 and NAX2, which work together in an interactive or epistatic way rather than just adding their effects. The identification of a molecular marker linked to the NAX1 gene has significantly facilitated the rapid transfer of this valuable salt tolerance trait into commercial durum wheat varieties through marker-assisted breeding, speeding up the development process considerably.
So it's not just about keeping sodium out entirely, but also sometimes managing what does get in and where it goes within the plant.
Exactly. For species or varieties that cannot effectively exclude salt from their water supply, especially under high salinity, a crucial cellular strategy is to compartmentalize the salt. They sequester it, storing it away safely inside large cellular storage sacs called vacuoles, thereby protecting the sensitive cytoplasm and organelles where metabolism happens. This sodium sequestration into the vacuole depends on specific protein pumps in the vacuolar membrane, the tonoplast. These include Na plus H plus antiporters, which swap sodium for protons, as well as proton pumps, V-type H plus AT passes and H plus D pases, that generate the necessary proton gradient to power the antiporters. As a compelling proof of concept, researchers created genetically modified Arabidopsis thaliana plants, a model species that overexpress specific wheat genes encoding these vacuolar pumps, either the proton pump, TBP1, or the sodium proton antiporter, TNHX1. These transgenic Arabidopsis plants were shown to be much more resistant to high concentrations of salt and also, interestingly, to water deprivation or drought conditions compared to their normal counterparts. This illustrates the fundamental molecular targets that can be modified, either through transgenics or potentially through induced mutation, to confer enhanced salt tolerance by improving the plant's ability to safely store excess salt away from sensitive cellular machinery. It's about making the plant much more robust in these challenging environments.
Okay, shifting gears from environmental stress to biological threats. Disease can absolutely wipe out entire harvests, making food security incredibly precarious. It's a constant battle for farmers. But induced mutations are providing new built-in defenses against devastating pathogens, making crops inherently more resistant from the inside out.
That's absolutely vital work. Let's look at okra again, for example. We mentioned it briefly before. Yellow vein mosaic virus disease, or YVMD, which is caused by a begomavirus transmitted by whiteflies, is the most serious factor affecting okra production in Thailand, impacting both exports and domestic consumption significantly. To combat this, seeds of susceptible okra varieties were exposed to gamma rays at specific doses, like 400 and 600 grays. Researchers then meticulously screened plants over multiple generations from M3 to M7 under both field and greenhouse conditions to find individuals showing resistance to the virus. The effort paid off. One specific M4 generation plant, derived from a variety called Okura B21, irradiated at 400 gray, was found to be highly resistant to YVMD. Importantly, growers who tested these mutant lines were reportedly satisfied not only with the resistance, but also with the plant's overall stature and fruit shape, and crucially, the delayed disease development. This provides a longer, more productive growing season and harvest window. If you were to imagine an okra field, a susceptible plant affected by YVMD would show those characteristic distinct yellow veins, maybe curling leaves, and stunted growth. In contrast, the resistant mutant would display healthy, vigorous green leaves and normal growth, a stark visual difference illustrating the success of this mutation breeding approach.
That's a clear win against a damaging viral threat. What about other important food legumes, like chickpea?
Chickpea provides another strong case study for disease resistance through mutation breeding. The large-scale cultivation of high-yielding and disease-resistant chickpea varieties, such as CM88 and CM2000 developed in Pakistan, has greatly helped to stabilize and even increase chickpea production in the country. Since their release and widespread adoption, no serious epidemics of ascochyta blight, a major fungal disease, have been reported, demonstrating the significant long-term impact of deploying these resistant varieties. CM88 is specifically noted as a multiple disease-resistant chickpea mutant variety, and CM2000 was evolved by inducing genetic variability in an imported variety using gamma radiation, 150 gray dose. It's now recommended for cultivation in both rain-fed and irrigated areas of Punjab province and has significantly helped increase the production of Kabuli-type chickpeas. Turkey has also officially released a chickpea mutant variety named Tak Saigul, developed through mutation breeding, reflecting global success in using this approach to combat chickpea diseases.
It sounds like induced mutations offer a broad spectrum of solutions to disease problems across many different types of crops.
They really do. Consider pineapple and bacterial heart rot disease. This is described as a most serious disease for susceptible pineapple cultivars in Malaysia. Researchers used acute gamma-ray irradiation on young pineapple plantlets grown in vitro in the lab to select for resistant mutants. Doses ranging from 10 to 40 gray were applied, and the resulting shoots were repeatedly subcultured up to the M1v5 generation. This helps to minimize chimerism, ensuring the mutation is stable. Interestingly, at the higher doses, 30 and 40 gray, they not only found resistant plants, but these resistant plants also showed a significant increase in total sugar content, giving a higher sweetness index, and also produced heavier fruits compared to the non-irradiated control plants. That's a fantastic dual benefit: disease resistance and improved fruit quality.
That really is a double win. What about a challenging tree crop like citrus? They can be slow to breed traditionally.
Citrus canker, caused by the bacterium Xanthomonas citri subsp. Citri, inflicts great economic damage worldwide, particularly on susceptible sweet oranges. To find a solution, researchers selected induced mutant clones of the sweet orange cultivar Para, based on their showing a lower intensity of canker symptoms after inoculation. Specific clones, designated 9.1, 9.2, and 9.3, consistently demonstrated a lower incidence of the disease over time. This resistance was characterized by having the longest incubation period before symptoms appeared, developing smaller lesion diameters, and showing a lower overall disease severity score, AUDPC. This work is highlighted as one of the first successful reports of using citrus-induced mutations specifically aimed at obtaining greater resistance to diseases like canker.
Now, what about the crucial banana crop? You mentioned earlier they often struggle with limited genetic diversity due to their propagation methods. Can mutations help there too?
Bananas are particularly challenging, yes. Most commercial cultivars rely on asexual vegetative propagation, essentially cloning through suckers or tissue culture. This leads to very restricted genetic variation within cultivars, leaving them highly vulnerable and with little inherent resistance to pests and diseases. Traditional conventional breeding is also severely limited by the fact that many cultivated bananas produce very few, if any, viable seeds, making crossing difficult. Induced mutations offer a way to create new variation within these existing popular cultivars. In Ghana, for instance, the irradiation of vegetative buds from cocoa varieties, though not banana itself, it shows the principle in a related crop, resulted in the production of a mutant variety resistant to cocoa swollen shoot virus, CSSV. On-farm trials with this mutant showed significant increases in yield by farmers, with plants showing no symptoms of the devastating disease. For black leaf streak disease, also called black sigatoka, which is arguably the most destructive disease of bananas globally, research has focused on understanding the genetic basis of resistance. Scientists identified over 50 distinct resistance gene analogs, RGAs, in a resistant wild banana relative, Musa acuminata calcutta 4. Molecular analysis showed these genes belong to a specific class, non-TIR NBSLRR, commonly found in monocot plants like banana. This fundamental molecular work identifying resistance genes lays the essential groundwork for future strategies, potentially involving mutation breeding or gene editing, to introduce durable resistance into cultivated banana varieties.
Okay, so the power of mutation isn't just about survival fighting off diseases and stress. It's also about unlocking entirely new possibilities. Things like making fruit seedless, creating stunning new ornamental plants, and even boosting yield in perhaps unexpected ways.
Indeed, the applications are incredibly diverse, moving beyond just defense. Take seedless citrus, for instance. That's a highly desirable trait for consumers. This has been achieved through gamma irradiation of budwoods from seeded citrus varieties. Survival rates vary depending on the dose and the specific citrus variety, but the process ultimately led to the development and release of promising seedless citrus mutants. If you look at images comparing original seeded varieties with their mutant XLN seedless counterparts, you can visually discern the clear absence of seeds and sometimes subtle changes in the internal structure or shape of the fruit, making them much more appealing and convenient for consumers.
That's a direct market improvement driven by consumer preference. What about staple foods like rice, but with a unique, desirable twist like aroma?
Aromatic rice in India presents a great example here. Short-grain aromatic rices, varieties like Kalanamak, Dibraj, Kalajira, are highly prized locally for their unique sensory characteristics, aroma, taste, texture, and specific cooking qualities. But traditionally, these prized varieties suffer from low productivity. They take a long time to mature, long duration, and they tend to grow very tall plant stature, which makes them prone to falling over, lodging, especially with fertilizer application. All this limits the economic returns for farmers. To address these shortcomings while preserving the valuable aromatic traits, a mutagenesis approach was attempted. Seeds from 12 popular aromatic cultivars were treated with different doses of cobalt-60 gamma rays. The selection process over subsequent generations aimed specifically for mutants that were erect, semi-dwarfs, shorter and sturdier, non-lodging, and had high yield potential, all while carefully ensuring they kept the unique grain type and the desired cooking and aromatic quality traits of the parent cultivar. It's a balancing act, and they found success. Mutants with approximately 20% shorter stature were derived from several of these traditional varieties. Mutants showing about a 10-day reduction in the time to maturity were also isolated. One specific mutant line, derived from Kalanock, showed a mean grain yield of more than 2.5 tons per hectare, significantly higher than the local average of 1.52 tons per hectare. It also had a beneficial narrow flag, leaf, and leaf angle, which allows for denser planting and potentially even higher yields per unit area.
So it's about optimizing for agronomic efficiency and yield while carefully retaining those unique, desirable quality traits. What about ornamental plants, where appearance, color, and form are absolutely everything?
Induced mutagenesis is a well-established and highly successful method for plant improvement in ornamentals. It's widely used commercially. One researcher mentioned in our sources successfully developed 76 new mutant varieties using gamma radiation in a range of different ornamental species, with a major focus on chrysanthemums. Extensive work has been done on optimizing the process for ornamentals, determining radiosensitivity, selecting the right starting materials, refining methods of exposure to gamma rays, finding the suitable dose range, effectively detecting mutations, which can be subtle color flecks initially, isolating the mutants, and eventually bringing them to commercial exploitation. Techniques like recurrent irradiation, multiple exposures, and combining radiation with chemical treatments like colchicine, which affects chromosome number, have also been explored. Really unique and novel traits have been generated this way. Completely changed flower types, the development of strange appendage-like structures on the florets, flowers with stripes or sectors of different colors, and the induction of unusual tubular florets instead of flat ones. The development of late-blooming varieties in chrysanthemum, for example, achieved through mutation breeding, has had a tremendous commercial impact by extending the marketing season. However, a main drawback, especially with vegetatively propagated plants like chrysanthemums, which are grown from cuttings, not seeds, is that mutations often first appear as chimeras.
Chimeras, meaning the mutation isn't in all the cells.
Exactly. It means mutated cells are present alongside normal, non-mutated cells within the same tissue, like the flower petal. This is a big challenge because it's like trying to get a pure red flower when only a few cells have mutated to red and the rest are still the original white or yellow. It makes it very difficult to isolate and consistently reproduce the new trait, and many potentially desirable mutants might be lost because the non-mutated cells outgrow the mutated ones.
So how do breeders overcome that chimera problem?
To overcome this, a novel technique has been standardized. It involves using in vitro culture, growing plant tissues in the lab to directly regenerate new shoots from tiny pieces of the flower petals, fluorescens plants, that show the desired mutation. This method bypasses a lengthy callus phase and undifferentiated massive cells, which can sometimes cause genetic instability, and it helps to isolate pure, non-chimeric mutant lines more efficiently.
That sounds like a sophisticated solution to a really tricky problem in breeding these types of plants. And what about less common, maybe underutilized crops? Is mutation breeding being applied there too?
Yes, even in underutilized crops, like a medicinal tuber called Solenostemon rotundifolius, also known as coleus potato or house of potato, induced mutation is being applied to improve it. The goal here was to improve this potentially valuable but neglected crop. Single node cuttings were mutagenized, and superior genotypes were then selected based on improved tuber yield and other yield-contributing characters. Another key selection target was photoinsensitivity, meaning the plants can form tubers well regardless of the day length, which is crucial for expanding cultivation to different latitudes or seasons. Through this process, 14 superior mutant lines were isolated and successfully tested, confirming their photoinsensitivity across multiple growing seasons.
Okay, and finally, let's just quickly look at some direct yield enhancements mentioned in a few other diverse crops. It seems to pop up everywhere.
It really does. In edible mushrooms, specifically Pleurotus species, oyster mushrooms, studies showed that both gamma radiation and UV radiation could induce mutants with significantly improved yield compared to the original strain. For example, one strain had an average yield of only 8.5 grams normally, but this dramatically increased to 126.0 grams after a specific dose of gamma radiation treatment, according to one table in the source. For taro, Colocasia esculenta, a tropical root trop, an in vitro mutation program was implemented primarily to improve resistance to a devastating fungal disease called taro leaf blight. Plants were exposed to gamma radiation, and an effective mutation dose, LD30 killing 30%, was determined. Molecular analysis like RAPD was planned to help characterize any resistant mutants found. For yams, Dioscorea species, another important tuber crop, gamma ray radiation was applied directly to the young tuber sets used for planting. Researchers noted that the most effective dose range for mutation induction must be carefully determined. It needs to be between dosages that cause significant, but not excessive, lethality, maybe around 50% survival, as one expert recommended, and growth reduction. Too high a dose kills everything. Too low doesn't induce enough mutations. And finally, in black pepper, a spice crop induced mutations led to the development of a mutant line, M127, which significantly out-yielded the standard Czech variety across all tested agroecological zones in Sri Lanka. For instance, yielding almost 33 tons per hectare compared to about 21 tons per hectare in the wet zone. Farmers also reported positive side benefits like fewer incidences of pests and diseases and good keeping quality of the harvested peppercorns, highlighting the potential for multifaceted benefits from induced mutations. All right, let's do another mega recap, this time for Section 4. We really saw the diversification of applications for induced mutations here. First, we explored how they enhance abiotic stress tolerance, making crops more resilient against environmental threats like drought and salinity, an absolutely critical need in our changing climate. Then we delved into how mutations build inherent disease resistance, serving as built-in defenses against devastating pathogens in a wide range of crops, from okra and chickpea to pineapple, citrus, and potentially banana. And finally, we saw the cultivation of entirely novel traits going beyond just survival, making citrus seedless, developing high-yielding aromatic rice, creating stunning new ornamental flower varieties, improving underutilized tubers like coleus, and boosting yields directly in diverse things like mushrooms and black pepper.
It's a compelling expansion of possibilities, isn't it? As you think about this wide array of innovations, maybe drive differently by considering how these advancements broaden the range of cultivable land, allowing crops to thrive where they once couldn't, and also how they truly enhance crop resilience against both natural pressures like disease and human-induced pressures like climate change. This isn't just about tweaking existing traits, it's about pushing the boundaries of what agriculture can achieve. Time for another flashcard check. Key terms from this section include abiotic stress tolerance, drought tolerance, salinity tolerance, and sodium exclusion sequestration, yellow vein mosaic virus, YVMD, Ascochyta blight, bacterial heart rot, citrus canker, cocoa swollen shoot virus, CSSV, black leaf streak disease, seedless citrus, aromatic rice improvement, ornamental mutations, the challenge of chimerism, in vitro direct shoot regeneration as a solution, general yield enhancement, and photoinsensitivity. Quite a list. V, the molecular frontier. Decoding and directing evolution. Okay, so the journey of induced mutations clearly started with observations in the field, right? Literally looking at what popped up in those gamma gardens or farmers' fields. But the real revolution, the ability to truly understand and perhaps even precisely direct these changes, is happening now at the molecular level, reading and maybe even rewriting the genetic code itself.
That's absolutely the molecular frontier we're on now. Advances in genomics, particularly with the complete sequencing of model plant genomes like Arabidopsis and rice, have been a complete game changer for mutation breeding. It's now possible to identify a particular mutation with incredible precision. We can pinpoint the exact size of a missing piece of DNA, a deletion, and the precise point of a single letter change, a point mutation, at the molecular level. This deep understanding also helps us unravel the complex mechanisms of gene function. What does this gene actually do? And also how genes are repaired after damage, connecting back to those HR and NHEJ pathways we discussed. For instance, studies in maize involving plants with an abnormal number of chromosomes, or parts of chromosome, segmental aneuploids, showed really complex gene dosage effects. A plant that was trisomic, had 3 copies instead of 2, for most of the short arm of chromosome 5 revealed something surprising. Only about 40% of the expressed genes located within that extra chromosome segment showed the expected 1.5-fold increase in gene expression. The other 60% were not altered significantly, despite having an extra copy. Furthermore, and perhaps even more surprisingly, 86 genes located outside the aneuploid region were found to be altered in their expression levels. This suggests that the visible changes in a plant's development, the phenotype, caused by these chromosomal abnormalities, might be related to these complex, unexpected patterns of gene expression induced across the genome, not just a simple effect of having extra copies of genes in one region. It's not always a straightforward one-to-one relationship. There can be a cascade of effects throughout the genome.
So it's about understanding that deep, hidden ripple effect across the entire genetic network. And can we actually see these larger changes like deletions or rearrangements at the chromosome level?
Yes, definitely. Studies in wheat, for example, have detailed the induction of structural changes in specific small segments of chromosomes derived from Hinaldia villosa, a wild relative of wheat known to carry useful genes like disease resistance. This was done by irradiating the mature female gametes, egg cells, before fertilization. Among the resulting M1 plants, the first generation after irradiation, many showed structurally changed chromosomes. Researchers identified various types of changes, including terminal translocations, where a piece attaches to the end of another chromosome, interstitial translocations, where a piece inserts into the middle, and simple deletion chromosomes, where a piece is lost. The frequency of plants showing these small fragment structural changes was remarkably high, around 18.3% in this study. These specific lines, with their precisely defined chromosomal rearrangements involving small segments from the wild relative, are potentially very useful materials for chromosome-based physical mapping. They help scientists map genes to very specific locations on chromosomes, almost like finding a specific house on a very detailed street map, which is crucial for breeding.
Okay, so we can see big chunks moving around. How does this level of detail translate down to the actual DNA sequence analysis? Can we see these changes right down at the single letter level, the SNPs?
Absolutely. Modern sequencing technology makes this feasible. In soybean, for example, researchers have employed high-throughput sequencing instruments, like the GSFLX system, combined with specialized DNA amplification techniques, like DOP-PCR, to conduct comprehensive surveys of single-letter changes in the DNA code, the single nucleotide polymorphisms, or SNPs, across mutagenized populations. In one study looking at an EMS mutagenized soybean population, they detected a remarkable total of 1,187 SNPs within the sequenced regions, giving a frequency of about 1 SNP for every 272 DNA base pairs analyzed. Imagine looking at lines of text representing the DNA sequence from different individuals. You would clearly see where individual letters, A, T, C, or G, in the code have changed from one plant to the next. Our source material even included an image showing a portion of these aligned sequences with arrows pointing out the detected SNPs. This provides an unprecedented level of detail for understanding the precise genetic variations induced by mutagenesis at the most fundamental level.
Right. So it's one thing to see a mutated plant in the field and say, that looks different. But it's entirely another thing to know exactly which gene, maybe which tiny single letter change, caused that observable effect. This is where genetic mapping, driven by all this molecular analysis, truly comes into its own, turning observation into precise knowledge, pinpointing the cause.
This is the essence of connecting genotype, the genetic makeup, to phenotype, the observable traits. It's fundamental to modern breeding. Take the development of the 6-row spike in barley, which we mentioned as a key agronomic trait. This characteristic, having 6 rows of grains instead of the ancestral 2 rows, is controlled by a single gene, often called VRS1. Scientists use sophisticated mapping techniques, essentially triangulating the gene's position using molecular markers to pinpoint its exact location on barley chromosome 2H. This is called map-based cloning. They discovered that the VRS1 gene encodes a type of protein called a homeodomain leucine zipper, HDZIP, transcription factor. These proteins regulate the activity of other genes. They found that this VRS1 protein was specifically produced only in the developing lateral spikelets, the ones that normally don't fully develop in 2-rowed barley during early spike development. This suggested that the dominant functional VRS1 gene normally acts to repress the development of these lateral spikelets. Therefore, a mutation that inactivates this VRS1 gene releases that repression, allowing the lateral spikelets to fully develop, resulting in the desired 6-rowed spike. It's like identifying the exact off switch for lateral spikelet development.
That's a perfect example of identifying the switch that controls a trait. What about that male sterility example in rice you mentioned earlier? How does mapping help understand that, especially its connection to hybrid seed production? Just to remind everyone, in plant breeding, sometimes we want a plant to be male sterile, meaning it can't produce its own viable pollen. Why? Because it forces cross-pollination with another desired parent line, which is absolutely key for creating hybrid seeds. Hybrids often exhibit hybrid vigor, leading to significantly higher yields.
Right. So a fascinating case involves a specific genic male sterile mutant in rice called MSH, which was induced by chemical mutagenesis. This mutant had what's called a pleiotropic effect, meaning the single gene mutation affected more than one observable trait. It not only caused complete male sterility, but also resulted in a visibly chalky appearance in the endosperm, the starchy part of the grain. Using map-based cloning again, scientists meticulously tracked this mutation through generations of crosses and mapped the responsible gene MESD to a specific location on rice chromosome 9. They then discovered that the mutation itself was a splicing error, a mistake in how the gene's instructions were processed, specifically at the boundary of the 14th intron of a gene called UDP glucose pyrophosphorylase 1. UGPASTE1. This enzyme is crucial for starch synthesis. Consequently, the endosperm of the mutant rice grain had starch granules that were more roundish and smaller than normal, and the overall emulsi-mylopectin ratio, types of starch, was altered, causing the chalky look. Our source even included an image showing the normal versus the male sterile rice plants, alongside a genetic map of chromosome 9 highlighting the precise location of the MH gene using molecular markers. Now, this pleiotropic effect, the visible link between the desired male sterility and the easily identifiable chalky seeds, allows for a clever hybrid seed production system. The relatively low density of these chalky seeds makes it easier to visually sort and identify the male sterile seeds within mixed seed lots before planting, helping to ensure the purity of the female parent line in hybrid production fields.
So a single mutation creates a genetic marker that's physically observable, making the practical side of breeding much easier. Are there other examples of deliberately combining different desirable traits, maybe like dwarfing and male sterility, into one neat package for easier breeding?
Yes, that's exactly what's been achieved with the development of dwarf male sterile wheat lines. This involves creating a new germplasm that tightly links a specific genic male sterile gene, MS2, originally from the Taigu discovery, with a potent dwarfing gene, RHT10, from the ABN1 variety, physically close together on the same chromosome arm, 40S. The linkage is so tight, only about 0.18 crossing over units apart, that the two traits almost always stay together during reproduction. This tight linkage means that, in segregating populations, the male sterile plants are consistently and visibly shorter than their male fertile siblings. This makes it incredibly easy for breeders to identify and select the male sterile plants based simply on their shorter height without needing laborious pollen checking. This dwarf male sterile wheat breeding system has been successfully created and is being used as a practical tool for recurrent selection and population improvement in wheat breeding programs. Several new wheat cultivars, like RS981, RS987, have already been developed using this innovative linked trait system, demonstrating how strategically combining mutations can significantly streamline breeding efforts.
That's a really clever way to visually select for a crucial breeding trait, just by looking at plant height. What about using a mutation defensively, maybe to identify and eliminate hybrid seed contamination, ensuring the purity of a valuable hybrid crop?
That brings us to the bentazon lethal mutants in rice we touched upon briefly. Two such mutants, ADO77S from gamma radiation and Norin 8 meters, also from radiation, are controlled by single recessive genes named BEL and BSL respectively. Further studies show these are actually allelic, meaning they're different mutant forms of the same gene. These mutants have a unique characteristic. They are killed at the seedling stage by the application of the herbicide bentazon, even at a dosage that is completely safe for their true F1 hybrids, which have one normal copy of the gene, and all other normal rice plants. They are also sensitive to some other herbicides, and this sensitivity is also controlled by mutations in this same gene. Both the BEL and BSL mutant genes were successfully cloned using map-based cloning techniques, pinpointing the exact gene responsible. Sequencing revealed that each mutant allele had a single base deletion in its DNA code, G deletion in BEL, C deletion in BSL, leading to a non-functional protein. The wild-type, functional gene was found to encode a novel cytochrome P450 monoxygenase enzyme, subsequently named CYP81A6, which is apparently involved in detoxifying bentazon in normal rice. Now, these bentazon lethal lines are utilized strategically in certain hybrid rice seed production systems, particularly the two-line system, which relies on environmentally sensitive male sterility. The problem is that sometimes, due to temperature fluctuations, the male sterile female parent line might accidentally self-pollinate. If seeds from this unwanted self-pollination get mixed in with the valuable hybrid seeds, it reduces the yield potential and uniformity of the farmer's crop. By using a bentazon lethal female line, any seedlings resulting from accidental selfing, which would be homozygous for the lethal gene, can be selectively killed by spraying the seedling nursery or field with bentazon, while the true hybrid seedlings, heterozygous, remain unharmed. This ensures the genetic purity of the hybrid seed lot sold to farmers. If you were to see a field where this system was used after accidental selfing occurred, you would observe patches of dead brown seedlings, the self-lethal ones, within the healthy green hybrid crop, clearly indicating where contamination happened and allowing for its effective removal. It's a genetic safeguard.
That is incredibly clever, using a lethal mutation as a quality control tool. Are there other elements within the genome itself besides the genes we target that might contribute to mutations and genetic variation? Maybe something less direct than radiation or chemicals?
Yes, that's an important point. The genome isn't static. Retrotransposons, for example, are a fascinating area of research in this context. They are considered a possible intrinsic source for mutations because they have the potential to move around and spread within the genome using a copy and paste-like mechanism. When they insert themselves into or near a gene, they can disrupt its function, causing a mutation. A study identified a new type of Ti3 gypsy retrotransposon family in cultivated allotraploid peanut, which has genomes from two different ancestral species, and also in its diploid wild ancestors. Using techniques like phage fluorescence in situ hybridization, which lights up specific DNA sequences on chromosomes, they showed that this retrotransposon element is dispersedly distributed across the peanut chromosomes. Interestingly, it seemed more prominent in the A genome chromosomes, one of the ancestral sets, compared to the B genome. The estimated copy numbers were quite high, ranging from about 830 to maybe 3,000 copies per haploid genome, depending on the species. Now, while the specific sequences they isolated contained multiple stop codons, suggesting they might not be actively jumping around anymore, their sheer presence and potential mobility highlight another layer of genomic dynamism. These jumping genes can be a natural source of mutation and contribute to the genetic variation that drives evolution and provides raw material for breeders, adding a further dimension to the complex story of plant genetics. Okay, let's pause for a micro recap here. In this last segment, we've really shifted focus from the observable impacts of induced mutations, the traits we see in the field, to the intricate molecular world that underpins them. We've seen how advances in genomics, like full genome sequencing and S&P detection, allow for the precise identification of mutations, even down to single nucleotide changes. We also touched on how understanding complex gene dosage effects provides deeper insights into why certain mutations cause the developmental phenotypes we observe. Then, crucially, we explored the process of pinpointing the specific genes responsible for mutated traits, using sophisticated map-based cloning techniques. This helped us understand the mechanisms behind traits like the six-rowed spike in barley, the combined male sterility and chalky endosperm in rice, useful for hybrids, the linked dwarf and male sterile traits in wheat, and even the herbicide-lethal mutants used for hybrid purity control in rice. We also briefly touched upon the potential role of mobile genetic elements, like retrotransposons, as another source of natural genomic variation and mutation.
It's truly a dive into the unseen machinery of life. As you, our listener, think about this molecular understanding, maybe try to derive differently by considering how this level of precision could accelerate future breeding efforts. Could it make the process far more predictable and efficient compared to the more serendipitous, chance-based methods of the past? It feels like we're moving towards accelerating evolution with much greater intent and knowledge. Now, what's next? Where does this molecular frontier lead, and what is the ultimate goal?
All right, where do we go from here? The ultimate grand goal for plant genomics and mutation breeding, arguably, is not just to find individual mutations or understand single genes, but to understand every possible consequence of every gene within an organism. It's an incredibly ambitious vision, often referred to as mapping the phenome, the complete set of phenotypes or observable traits produced by a genome. Arabidopsis thaliana, that small, unassuming flowering plant related to mustard, serves as a crucial model organism for pursuing this goal. Because its genome is small, its life cycle is short, and vast genetic resources exist, it's now theoretically possible, or getting close, to investigate mutant lines for almost every single one of its roughly 27,000 genes. This makes Arabidopsis a model not just for general plant biology research, but specifically for achieving complete saturation mutagenesis, having mutations available for every gene and enabling a total analysis of all genes and their corresponding mutant phenotypes within a single plant species.
That sounds like a monumental undertaking, mapping every possible genetic outcome to every observable trait. How far along are we in actually achieving this phenomenal project? Is it still just a dream?
It's definitely underway, though far from complete. The future goal is indeed to collect comprehensive information about the knockout type mutant phenotypes, where a gene is completely inactivated, for each Arabidopsis gene. Significant progress has already been made. Huge collections, thousands upon thousands of Arabidopsis lines containing insertions of known DNA sequences, like dissociation or D's transposons, have been generated. These insertions often disrupt the gene they land in, creating a mutation. Researchers have systematically observed the visible phenotype, what the plant looks like of each of these lines under standard growth conditions. They've classified hundreds of clear, visible phenotypes, things affecting size, shape, color, flowering time, etc., into dozens of distinct categories. These phenotypic images, along with their detailed classifications, have been entered into searchable public databases. Imagine going to one of these online databases. You could potentially search by a visible trait like dwarf plant or late flowering, and the search results would display images of the corresponding mutant plants, linking the visible trait back to the disrupted gene.
So it's about building this massive, comprehensive library, connecting genes to visible effects. What about phenotypes that aren't immediately visible? Things like biochemical changes or responses to stress.
That's absolutely part of the comprehensive analysis needed for a true phenome project. Researchers are selecting homozygous lines where both copies of the gene are mutated for these insertional mutants and subjecting them to a battery of tests to detect non-visible phenotypes. They're using various categories of measurement, physical, chemical, biological methods to search for diverse traits as part of this total phenome analysis. This explicitly includes investigating stress-responsive or conditional phenotypes, where a trait might only manifest under specific environmental conditions, like high salt, drought, pathogen attack, or nutrient deficiency. Furthermore, all available mutant phenotype information from published scientific reports is being systematically collected and integrated to create a truly comprehensive phenotype database of Arabidopsis genes and mutants, consolidating knowledge from labs worldwide. The ultimate aim, looking further ahead, is to potentially develop holistic spectral models of the phenome and of pleiotropy. This means understanding the full range of effects, both obvious and subtle, that can result from single gene mutations, almost like being able to predict all the possible ripples caused by one tiny change in the genetic blueprint.
That's a truly powerful vision for the future of plant science and breeding. But you mentioned earlier that some crops, those that don't produce seeds in the traditional way, like bananas or cassava, present unique challenges. How does this molecular frontier apply to them?
Yes, vegetatively propagated crops. Those grown from cuttings, tubers, suckers like banana, cassava, potato, yam, sugarcane present a distinct set of challenges for mutation breeding and molecular analysis. They typically lack botanical seeds for easy propagation and crossing. They rely on inducing somatic mutations occurring in the non-reproductive body cells for generating genetic variation. This leads to inherent issues we've touched on. Many are highly heterozygous, having different versions of genes already. The frequent occurrence of chimeras, mixtures of mutated and non-mutated cells, and linkage drag, where desirable mutant traits are genetically linked to undesirable ones, making selection difficult. Crucially, because the normal process of sexual reproduction involving meiosis, which shuffles genes and filters out major errors, doesn't intervene in their propagation cycle, it effectively bypasses the natural sieve mechanism that often removes lethal or highly detrimental chromosomal aberrations in seed-propagated crops. This can lead to the gradual accumulation of potentially high levels of unintended mutation events or genetic load over generations of vegetative propagation, creating a more complex genetic background to work with.
So what are the solutions for these important but genetically complex crops? How can we apply these advanced techniques effectively?
Researchers working on these crops strongly advocate for the judicious integration of validated biotechnologies alongside induced mutagenesis and other high-throughput forward genetics assays. This includes strategically using cell and tissue biology techniques like micropropagation or inducing temporary diploid stages to potentially achieve homozygosity for certain genes, to minimize or eliminate chimeras more effectively, and significantly shorten the duration needed to generate stable mutant lines. Additionally, modern molecular biology approaches, especially reverse genetics tools like gene editing, if ethically and regulatorily appropriate, and transcriptome assays, measuring gene expression patterns, can significantly enhance the efficiency and precision of the induced mutagenesis processes in these crops. The immense potential here, the grain of discovery we're pursuing, is to leverage this deep molecular understanding and these advanced tools to precisely direct evolution for crop improvement, making the breeding process more predictable and controlled, even for these challenging but vital vegetatively propagated crops.
Wow. From the geopolitical tensions of the Cold War, driving atomic research into unexpected places, to the intricate dance of DNA damage and repair mechanisms within a single plant cell, and from the success story of a single ruby red grapefruit to entire fields of drought-resistant crops thriving in harsh conditions. The story of induced plant mutations is a truly remarkable deep dive. We've really seen how human ingenuity, often spurred by necessity or unexpected historical forces, can tap into the fundamental processes of life itself to reshape our food systems, making them potentially more resilient, more nutritious, and hopefully more abundant.
It really is an incredible story of science and application. And this journey raises an important question, perhaps, for you, the listener, to ponder. As our understanding of plant genetics becomes ever more precise, and our ability to induce specific, beneficial mutations or even edit genes directly continues to grow rapidly, what new responsibilities and opportunities arise? How should we best shape the future of our food system and perhaps recalibrate our relationship with the natural world, given these powerful new tools? The implications for global food security, for sustainable agriculture, and even for how we define natural food are profound and worth careful consideration.
Absolutely. And this deep dive, as always, only scratches the surface. There are vast resources out there, like the official FAOIEEA database of registered mutant varieties, thousands of them. Each entry in that database represents decades of scientific endeavor, farmer collaboration, and persistent human curiosity. It's waiting for you to explore further.
Thank you for joining us on this exploration today. We invite you to continue your own deep dive into curiosity wherever it may lead you next.